Wounded epithelia must quickly repair themselves in order to maintain their essential barrier function. Tom Millard from the University of Manchester in the UK explains that epithelial cells bordering a wound respond by forming two types of actin-based structure: dynamic membrane protrusions that help the cells crawl into the wound and or seal it, and an actomyosin cable that encircles the wound and closes it like a purse string.
So between them, these two structures seem to be the key drivers of epithelial wound closure. What we don't know so much about is what's actually regulating the formation of these actin structures at the wound edge. So that was really our goal at the beginning of the study.
Led by postdoc Yutaka Matsubayashi, Millard and colleagues began by visualizing the very earliest stages of actin assembly after wounding Drosophila embryos with a high-powered laser.
One thing that we noticed, which hadn't really been spotted before, is that before we get the formation of this actin cable and the actin protrusions, we actually form these punctate actin structures at the points where neighbouring cells meet along the wound edge. And once these have formed, then we go on to form the more recognised actin structures. So we were quite curious to know what these structures were, because they formed so early, we thought they must have some kind of special role in initiating the process of wound closure.
First of all, though, the researchers investigated which regulatory proteins promoted the assembly of the different actin-based structures at the wound edge. They began by removing SCAR, an activator of the actin-nucleating ARP2/3 complex.
And we found that this had the effect of reducing the formation of the actin protrusions, but didn't affect the cable. So this suggested that SCAR is probably the principal nucleation mechanism for the actin protrusions, but not for the cable.
In contrast, removing the actin-nucleating formin protein diaphanous inhibited assembly of the actin cable around the wound edge.
Curiously, when we removed these genes, we actually saw very little effect on wound closure. This leads us to believe that there's actually quite a lot of redundancy in the process of wound closure. It's actually really quite a robust process, and it can cope with the loss of quite significant actin structures, and presumably in that case other structures can take over and close the wound.
Removing SCAR or diaphanous had no effect on the formation of actin puncta at the wound edge. Instead, assembly of these structures was regulated by the ARP2/3 activator WASP. Embryos lacking WASP failed to form actin puncta, but still managed to generate an actin cable and protrusions, indicating that, rather than being precursors of these later structures, actin puncta are distinct elements that could have their own unique function in wound healing.
One idea that came to us was that perhaps these are actually endocytic sites, because WASP has been previously implicated in endocytosis. So to test this, we looked at the localization of the endocytic regulator dynamin, and we found that dynamin nicely co-localized with these actin puncta, which led weight to this idea that these are actually endocytic structures, and that the wound edge is actually quite an active site of endocytosis.
Matsubayashi et al. Then examined the effect of inhibiting endocytosis, using either a temperature-sensitive mutant of dynamin or a dominant negative version of the endocytic regulator Rab5.
What we found was some really quite dramatic effects on wound closure. So firstly, actually the process of wound closure happened quite a bit slower when we disrupted endocytosis. And we saw a really quite dramatic loss of actin assembly at the wound edge. So this suggests, firstly, that endocytosis is required for wound closure, but more specifically that it seems to be important in regulating actin assembly at the wound edge.
Millard and colleagues thought that endocytosis might coordinate actin assembly by controlling the recruitment of actin regulators such as diaphanous.
We noticed that in our wild-type embryos, diaphanous accumulates at cell junctions around the wound edge once the wound's been made. And if we disrupted endocytosis, diaphanous was no longer recruited to the wound edge.
Millard and colleagues thought that intercellular adherens junctions might be a key endocytic target in this process.
We knew that the sites where we saw this accumulation of dynamin were places where the adherens junctions change morphology, so they undergo this remodeling process during wound closure. And indeed, if we blocked endocytosis, this remodeling didn't occur. So we looked for genetic interactions between dynamin and various components of adherens junctions, and we could find that we could enhance the dynamin phenotype by partial loss of adherens junctions components. So this led us to conclude that adherens junctions are in fact quite a key target of endocytosis during wound closure.
Finally, Matsubayashi et al. Examined whether endocytosis was also involved in healing mammalian epithelial wounds. When the researchers scratched a monolayer of mouse kidney epithelial cells, the cells failed to form either an actin cable or protrusions at the wound edge if endocytosis was blocked using the dynamin inhibitor dinosaur. Endocytosis therefore has a conserved function in epithelial wound healing, perhaps by remodeling the adherens junctions between cells at the wound edge.
So we think that adherens junctions are acting sort of like signaling centers which recruit actin regulators and coordinate actin dynamics at the wound edge. But in order to fulfill this function, they have to go a remodeling event in which they change structure and probably change composition. And this is the key role of endocytosis in this process. It's creating these kind of specialized signaling adherens junctions at the wound edge. Presumably, there's some kind of damage signal which is interfacing somehow with this junction remodeling process. So it could be a chemical signal, or perhaps it could be a mechanical signal. We don't quite understand what's happening there, so we're trying to figure that out at the moment.
In the meantime, however, you can learn more about how the endocytosis-dependent coordination of multiple actin regulators is required for wound healing in the paper by Matsubayashi et al., published in the August 3, 2015 issue of the Journal of Cell Biology.
You